There's only a few in this one, so thank you, Peter. All right, thank you for everyone for coming. My name's Julie, and I'm gonna tell a little bit about a story today called The Mechanisms of Ret-Mediated Invasion and Pancreatic Ductal Adenocarcinoma, but I'm gonna call it PDAC for short from now on because it's a mouthful. So let's start with what exactly is PDAC. So PDAC is the most common type of pancreatic cancer, makes up about 90% of cases, and it usually originates from either ductal epithelial cells or acinar cells. So if we zoom in on the pancreas here, we have the ductal epithelial cells lining the duct of the pancreas, and then acinar cells here, which are in charge of secreting the digestive enzymes that go on to the digestive system. This makes this type of pancreatic cancer exocrine in nature, in contrast to other pancreatic cancers that are a little bit more rare and that originate from cells that are hormone secreting cells, so more neuroendocrine in nature, like cells in the isolates of Langerhans. In terms of the state of PDAC in Canada, it's actually the third leading cause of cancer-related death in combined sexes. So when we look over here at incidence specifically, it's down lower at 9th and 8th place for males and females respectively, but then this shoots up to 4th for the leading cause of mortality when we stratify by sex. And this has to do with the minimal or the really low 5-year survival rate of around 10%. Sometimes it's lower. It's different in a lot of literature, but it is amongst the lowest of all cancer types. And unfortunately, there has been minimal progress over the past 20 years in terms of trying to increase this 5-year survival rate. So this points to a need for earlier detection methods for this aggressive type of cancer, which I'm sure Sarah will talk about later on this year. But then my research focuses more on identifying novel therapies to help address this aggressive cancer. So why is PDAC so lethal? One of the main factors, it's multifactorial in nature, but one of the main factors is that it has minimal early onset symptoms. So it leads to very late stage detection. In fact, a lot of patients, once they do present with symptoms, they're already at stage 4. And 80% of the cases exhibit either regional spread or organ metastases upon diagnosis. There are 4 major ways in which PDAC cells can disseminate. Um, the adjacent organ invasion, one of the most common areas is the small intestine. There's vascular invasion, which we've all heard of, lymph node spread, and then one mechanism that's a little bit less known, which is called perineural spread, where tumor cells actually disseminate along nerves. So all 4 of these mechanisms lead to early tumor dissemination and micrometastases. So even when these primary tumors are resectable, unfortunately, there is high recurrence rate after surgery. So a lot of these patients, even if the surgeons are able to get out as much as possible of the primary tumor, and if there are clear margins, there's a high chance that there's actually micrometastases somewhere else. In terms of perineural invasion, I like to call it a cool mechanism. It's not very well known, but it's when cancer cells actually infiltrate and disseminate along the peripheral nerves. And this happens in about 70 to 90% of PDAC cases, so quite a few. It contributes both to lethality and issues surrounding quality of life in patients. So the first aspect is that it seems to cause a decreased response to treatment. Some literature points to the fact that these cancer cells are able to hide, so to speak, within the nerves and stay away from systemic treatments. Then another aspect that I want to bring up that's often overlooked is that it actually causes extreme abdominal and nerve pain in these patients. So unfortunately, for the limited amount of time that they are alive, they experience quite a bit of pain that is often treated with opioids. So this points to, I guess, or underscores a need to not only find treatments to halt perineural invasion to increase treatment responses, but also just to address the quality of life in these patients. When we look at mechanisms of perineural invasion, we can look at it first as a holistic view. It's a complex where there's a complex interaction and reciprocal exchange of signals between tumor cells themselves and the peripheral nerves. A PhD student in my lab, Larissa, is looking at this reciprocal exchange and interaction more in depth versus my project, which focuses more on the structures and the invasion that's happening from the tumor cells themselves, which facilitates the original invasion towards peripheral nerves. So as I said, it's a complex interaction between tumor cells and nerves where several chemoattractants are actually secreted out into the tumor microenvironment. Some of these are neurotrophic factors, and relevant to my project is glial-derived neurotrophic factor, like GDNF, or other GDNF-like ligands or family ligands. And it just so happens that GDNF and other GFLs are the ligand for the RET receptor, which is the main receptor that our lab studies in terms of cancer. When we look at the RET receptor, it's a receptor tyrosine kinase, and in normal physiology, it's important for the development and the maintenance of enteric nervous system, the kidneys, and other neural crest-derived tissue during development. Activation of RET usually occurs, or often occurs, see the laser pointer here, upon binding of GFLs. So in this figure, we have GDNF specifically to their co-receptor, GFR alpha. Here we have GFR alpha 1. This leads to receptor dimerization, autophosphorylation of intracellular residues, and then interaction with mediator proteins to promote downstream signaling. Of note from my project, I want to bring up here is Stark kinase specifically, which has been shown to interact with RET. And this goes down to promote things such as focal adhesion formation, as well as signaling through the PI3K AKT pathway. RET signaling, when apparent, has been associated with tumor-promoting processes in a bunch of different cancers. Specifically, RET can act as an oncogene when there are mutations that result in gain of function. So this happens quite often in certain types of thyroid cancers, as well as some non-small cell lung cancers. But then there's also the fact that RET can act as an oncoprotein when it's just simply increased wild type expression, so more of the receptor on the surface of the cell. In PDAC specifically, it's expressed in about 40 to 65% of cases, so around half, but depending on which literature you're looking at. And it seems that elevated expression actually correlates with higher grade tumors and poorer prognoses. So this points to the question, does RET activity promote PDAC progression and these aggressive phenotypes? My project will look specifically at its role in cellular outgrowth formation as well as invasion, while there are other tumor-promoting processes as well associated with RET signaling, such as perineural invasion, proliferation, survival, and metastasis as well. So to look at RET's role in PDAC and invasion specifically, a previous PhD student in our lab named Dr. Eric Lian used shRNA RET knockdown cell lines, and he actually found that RET activation promotes both cell invasion and the formation of proteolytic structures. So here we have a 3D spheroid of pancreatic cancer cells or PDAC cells, and we see that when there's total RET knockdown, shRNA knockdown, in the presence of RET's ligand, there's actually decreased invasion into a surrounding matrix, so 3D, compared to when RET expression is present and activated with GDNF. We see a similar pattern here when we look at the single cellular level with proteolytic structure formation. So indicated by these darker patches here is where the cell was able to degrade collagen. And on the right, with the total RET knockdown, there are far less of these degraded areas compared to when RET is present and activated. So these proteolytic structures are actually termed invatopodia. And Eric also found that in the presence of RET's ligands, so GDNF, RET actually colocalizes or associates with another marker of invatopodia called TKS5. So you can see here that in the presence of GDNF, there's white spots. So that's the colocalized or the association of the red, in which depicts RET, and the kind of grayish puncta, which depict TKS5. So this points to perhaps RET-mediated invasiveness in PDAC involving the promotion of invatopodia. So what are invatopodia? If we look a little bit deeper, they're actually proteolytically active cell extensions. So I think they're really cool. They differ from other cell extensions like lamellipodia and filopodia in that they're able to degrade the extracellular matrix around them through the release of matrix metalloproteinases. Often MMP2 and MMP9 are associated with this process. Invasive formation is usually initiated by pro-invasive signals. So there has been past research showing that both EGFR and MET signaling are associated with increased invasive formation. So the rationale behind this is perhaps maybe RET signaling and activation could be another mechanism promoting this factor. And then I just want to highlight TKS5, which I mentioned on the previous slide. TKS5 is a scaffolding protein or adapter protein with 5 SH3 domains, so it's able to bind several partners at the same time. It's not a kinase, and it doesn't elongate the actin filaments, but it kind of acts as I would call it like the glue where other actin nucleation factors and adapter proteins can bind to it. Interestingly, TKS5 is actually phosphorylated by Src kinase, and as I've mentioned previously, Src interacts with and becomes activated downstream of RET. So this brings me to my hypothesis that RET activation promotes PDAC cell movement and the formation of invasive necessary for invasion. But to address this hypothesis, my first aim was to just generally explore both RET and TKS5's contribution to PDAC cell migration and invasion. To do so, I used two different cell models dependent on the assay, and I'll go into a little bit of detail about that now. The first PDAC cell model is called MIA-PaCa2 cells. On the left here, we have the wild type cells, and in the middle, we have a RET knockout generated using CRISPR-Cas9 technology by Dr. Matthew Krupe. And these cells are derived from a primary tumor, specifically an older adult male. They have a highly motile phenotype and a doubling hour of around 48 hours. And the reason we use this PDAC cell line is because it endogenously expresses RET. So we can see here the wild type has RET expression, while the RET knockout does not. More recently, or most recently, I guess, I was able to generate a TKS5 or TKS5 knockout using CRISPR-Cas9 technology with the help of a student named Yayun Tan. So I haven't done many assays with this yet, but looking forward to it in the near future. The other type of cell model that I use is called PANC1, and this is the cell model that I use for the majority of my assays. As you can see down here, it expresses relatively more RET compared to the MIA-PaCa2 cell line. And it's also derived from a primary tumor, this time a female, older adult. They move as a more collective unit. These cells are a lot flatter, and they have a longer doubling time. So in terms of assays like let's say collective cell migration with wound healing, it's more likely that they're collectively moving together versus just proliferation filling the wound. And then I also generated a CRISPR-Cas9 TKS5 knockout with another student named Dax. And thank you, Brandy, for generating the RET knockout, where we can see here again, RET expression was depleted. So to look at RET's role and TKS5's role in PDAC cell, just movement and functional cell processes. First, I looked at single cell motility using the MIA-PaCa2 cells because they are highly motile cell types. I then looked at the ability for the cells to move as a unit, so collective cell migration using wound healing. And then finally, we looked at transwell invasion. So are these cells able to degrade a barrier, specifically in this case, Matrigel, and move towards a chemotactic or a chemoattractant? So when we look at single cell motility, we actually found that RET activation does promote this process. So this graph here depicts mean squared displacement, which is the area in which the cells moved over a 12-hour time period. So we used a device called the InfuSight Zoom, which took an image of the cells that I plated out every 15 minutes, and then we created a sort of video and tracked the cell movement over the 12 hours. So this line up here depicts the wild type cells, which endogenously express RET in the presence of RET's ligand GDNF. And then below we have less movement with the wild types in the absence of RET activation. And then finally down here with the RET knockouts, so no expression of RET plus and minus GDNF, we have the least amount of movement. We can see this further here with these spider graphs, or spider plots, I guess, where we have each line depicting or representing the path of an individual cell over the 12-hour time period. So it starts at 0, 0, which would be its first coordinate, and then movement outwards from that. We can see the wild types minus and plus GDNF. The plus GDNF definitely visually just have more movement than the wild type minus, along with the RET knockout cells. And then finally, just to visualize this a little bit more, I have a quick little video of the wild type cells plus GDNF, so with RET activation, versus the RET knockouts plus GDNF. So I'm not sure if you can see this, but this is a common observation with these RET knockout cells, is that they'll form these focal adhesions or projections and try to move, but not be able to fully turn them over. Where up here, we have quite a bit of movement between the cell and then one coming in as well from the top. So this is looking at single cell motility, but what about the ability for these cells to move together? So next we looked at collective cell migration using the PANC1 cells, and we looked at 3 different cell lines, the wild types, which endogenously express RET, we had the TKS5 knockouts, and then comparing to the RET knockouts over a 30-hour time frame. And it's important to note that this is all 3 of these timelines, I guess, were done with the addition of GDNF. So there was a GDNF treatment. We can see here on the right that when we look at percent wound remaining, so 100% would be the first the area of the wound at the beginning, versus after 30 hours, the wound is, if the bar is lower, that means that the wound is closing more because there's less wound remaining. And we can see that when we knock out both TKS5 and RET, there's considerably less or decreased collective cell migration versus the wild type plus GDNF. When we stratify this further, when we look at a specific cell line, so here we have RET inhibition in wild type cells alone, and we have a vehicle, a GDNF treatment, and then a sulpercatinib treatment. So sulpercatinib is a selective RET inhibitor. It's FDA and Health Canada approved and indicated for non-small cell lung cancers with RET fusion proteins, along with certain thyroid cancers with RET mutations. And so when we see here is that when we add sulpercatinib, so inhibit RET, is also chemical inhibition of RET actually also decreases collective cell migration over the 30-hour time frame compared to the GDNF treatment here, but not compared to the vehicle. So that's without RET activation. You may be wondering, what about in the TKS5 knockout cells? So that's what we looked at next, is again with the vehicle versus GDNF, so RET activation and sulpercatinib. We found that once again, sulpercatinib was successful at decreasing collective cell migration, even in the absence of TKS5. So this points to perhaps RET being a greater or playing a greater role in collective cell migration than TKS5. As you can see here, after 30 hours, the GDNF compared to the vehicle showed a decreased wound percentage and then the sulpercatinib kind of made it so that there wasn't as much collective cell migration, so more percentage of that wound remaining. Finally, in terms of cellular assays, I wanted to look at the effects of RET and TKS5 knockout on cell invasion. So to do this, I used a transwell invasion assay, where on the top we plated the PDAC cells, specifically here the PANC1 cells, and then on the bottom we had either arterial smooth muscle cells or an HEK control. So the difference between the two is that the SMC cells secrete GDNF and other ligands, so it creates this constant chemoattractant gradient, which remains there over the 24-hour period, versus HEK cells, which do not secrete the GDNF, so RET's ligand. And then after 24 hours, we quantified how many of these cells were able to degrade through the matrodome matrix and down into the bottom and through the pores of the top insert. When we look at 2D cell invasion, just comparing SMC cells versus HEK cultures, you can visibly see that the cells that were co-cultured with SMC cells, were visibly able to invade much more than the cells that were co-cultured with HEK cells. This is what we expected to see, seeing as though SMC cells do secrete a lot of different growth factors, including GDNF, arterial smooth cells. But when we look and stratify this based off of just SMC cells in general, we have the wild type PDAC cells, the TKS5 knockouts, and the RET knockouts. And then we also wanted to see, okay, how much of this invasion is actually RET mediated in the wild type and the TKS5 knockout cells. So again, we have a sulpercatinib control here to inhibit RET signaling. And what we found is that TKS5 knockout cells invade less than the wild type cells with no treatment. So here the black is the wild type, and then the pink being the TKS5 knockouts. And then RET knockout cells exhibit decreased invasion overall. So with the no treatment, the vehicle, and the sulpercatinib treatment, they remain around the same. But then interestingly, when we add RET inhibitor sulpercatinib, this reduces wild type and TKS5 cell invasion down to the level of that of RET knockouts. So we can see here that RET definitely does have some sort of, or plays some sort of role in invasion in these cells, which was really interesting. And TKS5 does to an extent as well, but again, with inhibiting RET on top of that, it brings it down to further for invasion. To complement this, I then looked at epithelial mesenchymal transition markers in two cell types, the TKS5 knockouts versus the wild types. So in terms of mesenchymal markers, I did RT-qPCR and looked at encoherin and vimentin, which are both markers of more mesenchymal phenotype. And we can see that in the wild type cells, there's about double the amount of encoherin and vimentin compared to the TKS5 knockout cells. So this kind of complements what we saw with wild type cells being able to invade more than the TKS5 knockouts. And then on the flip side, when we look at epithelial cells markers like ecadherin and occludin is a cell junction, we see protein, we see that the TKS5 knockouts, it flips. They have more or higher expression of ecadherin, considerably more of ecadherin, and about double the amount of occludin as well. And this observation kind of matches with what you see in terms of just general phenotypes with these cells, where in the wild types, it's easier to get the panc cells off on their own, and there's a mixture, I guess, of these flatter cells versus cells that look like they would be more motile, versus the TKS5 knockouts, they generally grow in this cobblestone-like format. They're very flat, and it's extremely difficult to try and seed them off on their own. So future directions for my first aim. First, I'd like to explore TKS5 and RET's effect on directional single-cell motility. So previously, I plated the cells and just added GDNF as a treatment in general. This time, it'd be nice to have a source of GDNF to see that the cells actually move directionally towards them. And then also, I would like to add a RET-specific inhibitor like sulpercatinib to this to see if that impedes single-cell motility. Next, I would like to look at RET's role in 3D cell invasion by imaging PDAC spheroids embedded in collagen. So although Eric did this in the past, he did it with shRNA knockdown, RET knockdown cells in PDAC. Yeah, in PDAC cell lines. I'd like to do this with our RET knockout, our TKS5 knockouts, and again, look at RET inhibition. And this is what I'll be working on in Boston as well. We're doing some co-cultures, in fact, with some cells that express RET and some cells that do not, and see their propensity to move towards GDNF. This brings me to my second aim, which is to characterize RET and TKS5's role in PDAC invasive formation specifically. So now that we know that RET and TKS5 look like they play a role in promoting invasion, what about the mechanism behind this? So this is just another schematic that I kind of created showing RET activation and then TKS5. I want to point out cortactin here, which is another invasive marker that's important for recruiting actin nucleation factors, which are the factors that elongate the actin chains. And then MMP14, which is a membrane-bound matrix metalloproteinase that cleaves soluble MMPs, such as MMP9 and MMP2. So first, I wanted to see if RET and TKS5 colocalize, so further explore this relationship. While previously, Dr. Lian found that they associate in the same area, he was not able to quantify this, so I took a step back, and that's the first thing that I did. And I found that in the addition of RET's ligand, so in GDNF, is added, it does increase RET and TKS5 colocalization. So here we have staining for TKS5, staining for RET, and then a merged image. And wherever you see yellow here, and this is a script for colocalization, this picks up areas where there is overlap between the red and the green channels. So you can see here, when there's no GDNF, it actually has a negative colocalization coefficient or piercing coefficient, versus when we add GDNF, they colocalize more. Oh yeah, I don't know if it'd be better to see with no lights on, but thanks. Yeah, and anywhere outlined in white shows areas of colocalization picked up by the script. So this brings us to a very interesting observation between RET and TKS5, where we actually found that TKS5 depletion seems to reduce RET protein levels. So using our CRISPR-Cas9 TKS5 gene knockout, we can see here that in the wild type, which expresses TKS5, and then the TKS5 knockout, TKS5 is depleted. And with 4 separate replicates, we actually found that there are reduced levels of RET protein level in these TKS5 knockout cells. And you may think, okay, is this an artifact? Is this perhaps it's something secondary to the method that we use with the CRISPR-Cas9 technology. But if we go back to 2022 and look at Larissa's work, we see that when we used a lentiviral shRNA TKS5 knockdown, so a completely different method of bringing down TKS5 levels, there's a very similar observation where RET levels also decrease considerably in the TKS5 knockdown. So looking further into this, we wanted to see if it originates at the transcript level as well. So using RT-qPCR in both the TKS5 knockouts, and then thank you, Larissa, for doing it previously in the TKS5 knockdown cells versus their controls. We have on the left here, there are less transcripts being made of RET in the TKS5 knockouts compared to the wild type. And then when we look at panc TKS5 knockdowns and Miopaca, so a different cell line TKS5 knockdowns, there's a similar observation with decrease about 50% of the transcript being made of RET in the TKS5 knockdowns compared to the empty vector controls. The next thing I wondered was, is this a global phenomenon? So is knocking down or knocking out TKS5 also affecting other protein levels? So I looked at specifically cortactin and MMP14, as these have been shown to associate with TKS5 previously. But what I actually found is the opposite, where cortactin levels in the TKS5 knockout seem to be increasing. And this is a trend that's been observed with MMP14 as well. I only have 2 replicates of this, but both have been consistent in terms of increased levels of cortactin and MMP14. So although the levels of these proteins are increasing, does that actually translate to them being in the same area at the same time? So right now I'm optimizing another IF experiment where I'm going to look at, does knocking out TKS5 and RET affect MMP14 and cortactin colocalization? So this would be kind of a pseudo for an area where there's an invadopodia forming. So here we have IF of actin, so stained using fluorine, MMP14, and then down here we have cortactin and a merge. And you can see these areas here near the nucleus where cortactin has been or is colocalizing, or at least associating with actin. I'm still in the initial stages of this, but I thought it was a really nice picture to show. And I want to investigate RET's role in this process as well. So I'll have wild type cells, either plus or minus RET's ligand, the TKS5 knockout cells, and then RET knockout cells as well. Just for context, this is a TKS5 knockout cell, or 2, I guess, in the presence of RET's ligand, so RET activation. This brings me to my second aim and the future directions. The first are to discover whether there are changes in RET transcriptional regulators in the absence of TKS5. So we see that knocking out or knocking down TKS5 does affect RET transcript levels, so there are less. But is there a change happening to maybe perhaps the transcription factors that are regulating RET transcription in the first place? We can do this using RT-qPCR again, looking at different transcription levels, transcription factor levels. And then the second area that I want to do more research in is is there actually active degradation happening at these areas of colocalization? So just because two markers of invatopodia are colocalizing doesn't necessarily mean that they're able to actively, or that it's producing a structure that's able to actively degrade. So we can do something called dye-quenching collagen, or an alternative is FIT-C collagen, where when this collagen is degraded, it emits a certain fluorescent signal. So it'd be interesting to see if colocalization of these markers happen at areas of collagen degradation and whether knocking out RET and TKS5 impedes this process. Finally, just kind of look at the difference between MMP2 and MMP4 activation in the three different cell lines using gelatin zymography. So we have on the right here a previous study that did so, where we have the pro and active, so cleaved version of each MMP. So MMP2 and 9 are known as collagen and gelatin degraders, whatever. Gelatin is just a hydrolyzed form of collagen, so it's smaller form. So it'll be interesting to see if the activity of these MMPs is different between the wild type, the TKS5 knockout, and RET knockout cells. Finally, this brings me to the significance of my project. I'm hoping to establish mechanisms of RET-mediated invasion in PDAC and to better explore these cellular mechanisms. So I'm looking to perhaps implicate RET-specific inhibitors as a potential therapy for RET-expressing PDACs. Of course, these have to be further validated using in vivo models, but then after that, perhaps it may be, and after Larissa's work, it may be something that helps impede perineural invasion, which would be really interesting. And then also, resistance to small molecule inhibitors is a persistent issue in treatment of different cancers. So I thought that this is another way to kind of identify alternative targets in this pathway, including TKS5, which looks like when you knock out TKS5, this also affects RET. So that was an interesting add-on. But yeah. Thank you so much to my supervisor, Dr. Lois Mulligan, my committee members, Brandy, shout out Brandy, and then all my lab members. Yeah, thank you.
Wonderful. So questions, folks? Who's going first?
No.
Marco.
Sorry. Great job, Julie. This isn't a question about your data. I'm just a little bit curious, but
Yeah.
does the treatment of pain, like with opioids and stuff, modulate perineural invasion in any way?
It doesn't modulate perineural invasion in any way. No, it's just to alleviate the pain of the symptoms, yeah, associated with the symptoms. I actually have a hepatobiliary and like pancreatic neurosurgeon, not neurosurgeon, but surgeon on my committee, and he says, yeah, that the opioids are just last resort to make them feel the least amount of pain as possible. Yeah.
Yeah, I had the same thought here, but it affects the signaling in the neuronal cells in a way that might come out around. Other questions?
Yes, cool.
Are there any biologics that have been more successful than the small molecules, or the success rate is kind of certain?
Are you talking about like targeting RET specifically?
Yeah.
I only know of two different small molecule inhibitors right now for RET that are FDA and Health Canada approved. There are some other ones in trial that are actually like dual RET and SARC. I haven't seen any like antibody conjugates or anything like that. But yeah, just know of the small molecule inhibitors so far.
You guys can derive a precursor gene known as IGM-1.
Pinin.
Have you come across any data or anything that in case maybe Rett plays a role in IPM and that transition?
Yeah, no, that's a great question. It actually seems to happen later on as so not a driver mutation, but kind of just a passenger that comes on afterwards when these things start become or when these lesions start becoming more invasive. So it's more like the TP53 and then the KRAS, CDKN2, like, you know, all that. And then Rett later on, increased expression later on. Yeah, no problem.
Thank you.
Hey, Chris.
Just from your movie at the beginning, especially the one.
Do you want to go back to it?
You can put it back if you want. It's a nice one to show. The one cell in the knockout that seemed to originally, at least in my mind, they wanted to move, but didn't seem to let go at the back end and then.
Yeah.
Said, okay, I'm stuck.
Mm-hmm.
So. Are there effects on detachment?
Yeah.
Those processes that are impacted by red signal?
That's a great question. So my project looks specifically at invasion and invadipodia formation, but another cell extension or just cell structure in general are called focal adhesions. So these are like the little, if you think of a frog and the little pods on the end of it and how it like a cell sticks to whatever medium. Here it's plastic, but Rat and Stark specifically have been involved with or implicated in focal adhesion turnover. So my thought, and that's another avenue that you could go down, is looking at focal adhesion formation and how Rett inactivation can impair that. So I think that this is more of a focal adhesion pathway happening and lack of focal adhesion turnover versus my other functional cell assay is looking at invasion and invadipodia. Yeah. It's cool. It's like bobbing back and forth. Sorry, yeah?
A couple of questions if I could. Could you show your picture of the invader podia?
Oh, yeah. It's in a bunch of different places, so hopefully I'll just.
Yeah, this one here. So it looks here as though TKS5, which I've never heard of, is so it's a scaffold protein. It's involved in the formation of invader podia. How does that influence mRNA levels of Rett? I mean, that's what it's obviously, and then you're going to look at transcription factor levels.
Yeah.
But this is located normally out in the cytoplasm, maybe even on the membrane, is that right? So where is it and how is it involved in Rett stabilization or not?
Short answer is I don't know, but yeah, so you're right. TKS5 is not located within the nucleus whatsoever. It's in the cytoplasm, and then once Stark is activated, TKS5 has a domain, a PX domain, so which brings it to binding the cell membrane. But I have seen some things saying that TKS5 may have a role in vesicular formation and recycling of certain molecules. What's interesting is that I also checked the protein levels of other receptor tyrosine kinases, and I have that further down here, but it doesn't seem to affect, like I have EGFR and MET levels, and I've done it a few times. It's just a general little, but you can see here, so there's lower Rett in the TKS5 knockout, but MET and EGFR don't seem to be impacted. But I have seen some papers show EGFR also associates with TKS5. So I'm not sure what's going on there exactly. I'm going to look further into whether regulators of Rett transcription, like it might be a global effect where TKS5 being knocked out or knocked down is just impacting the levels of those transcription regulators specifically. But yeah, so that's the next step, is looking at that. If you have any ideas, I'd love to. I'm very open to trying to work through this, what's happening. Yeah.
I'll ask you another unrelated question.
Yeah.
So you show us lots of beautiful data about Rett and these mechanisms of motility, but I didn't hear anything that said about perineural versus invasion of other vascular invasion and lymphatic invasion. Do you think this mechanism or the mechanisms you're looking at are relevant just to the perineural invasion as opposed to the other forms of the way that tumors move?
Yeah, no, great question. I think it's more relevant to perineural because these neurons also express RET and they secrete RET's ligand versus if you think about, I guess some smooth muscle cells lining the vessels may secrete, but not to the extent that these nerves that are actually infiltrating within the area do. So we've seen before with co-cultures in the nerves that these cells that express RET and these nerves that express RET, they just love each other. Like they move towards each other like no other. I have not tried with like we haven't tried any co-cultures with like looking at lymphatic system or anything like that. But because RET is expressed like highly on these peripheral nerves and on these cells, is kind of just oh ding, let's see if there's a mechanism there or a reciprocal reaction there. Yeah, thank you.
See, you've looked
I don't know how to
transcript
change it.
levels in many cases, but also protein levels. So this would be a good example where you see situations. Do you always see correlation when transcripts go up, protein goes up, vice versa? Or have you seen in any case the opposite of that, where transcript levels are going up, but protein is not, or maybe even continues to go down?
I haven't seen that yet. When we go back here with MMP 14, I have looked at transcript levels of MMP 14 in the TKS5 knockouts. I've only done it once, so I didn't want to put anything up here that I've done it once, but it looks like the transcript levels are also increased as well.
And the same thing with Cortactin.
Cortactin, we don't have primers for, we're ordering them right now, but I'm would hope I'm
But
guessing
that
I see the same
transcript
thing.
levels go down.
Yeah.
Okay.
Yeah, at first we thought, oh, maybe it has something to do with ret turnover. So I'm gonna do
Yeah, that's the
cyclohexamide
reason.
assay. Yeah.
Protein is, for some reason, it's not stable. It's not engaged, and so stability sometimes is affected by
Mhm.
failure to engage. And so with TKS5 being an adapter protein, you wonder about that. And so it would have been very intriguing if the transcripts for RET were actually going up.
Yeah,
Because
to try and
sometimes
compensate.
cells are trying to replace the protein that they're missing or the function that it's providing for them.
I'm still gonna do the cyclohexamide, so degradation assay to see if that's something that's going on. But
Half-life.
yeah, half-life.
Yeah,
But
that would be. And I was intrigued by so your coherent, you did that at the protein level, the coherence and incoherent. Did you do it at protein and RNA level?
no, not protein, just started with RNA. We have a panel, an EMT panel, so I run that and see. I haven't done any EMT transcription factors yet though, like twist or anything like that, or have I? Can't
Because, the way your cells, the cell migration is affected is exactly what you would expect, that they're flipping to N, or they're flipping from a more of an N coherent phenotype to
An
an
E-coherent.
so-
Yeah.
and
Yeah, there's a lot more you could hear in the TKS5 knockout cells. Like here we've got like 1 versus 0.5. It's
they don't
consistently
seem
above.
to, in that movie, they don't seem to be flattening out and grabbing hold of one another though. They
No.
don't feel
Those are the Miopaca cells, so the more motile PDAC phenotype. I've only done the panel in the panc cells, which are in general flatter cells and don't move on their own. Yeah.
And so as far as the the 5-SH3 domains of this protein, has anybody figured out what their favorite binding partners are?
Cortactin is a big thing. Like TKS5 does bind cortactin. We're not sure if it binds ret or not because ret's more associated with binding SH2 domains, not SH3. There's a TKS4, which only has 4 SH3 domains. It's on a completely different chromosome, like it's a completely different gene. I want to look at levels of that too and see what's going on there. So, but yeah.
Any other questions? If not, join me in thanking.